Okay, I'll go ahead and get started. So we haven't met for about months, and a lot of exciting stuff has happened. If you follow us on social media, you know that we went to France this summer and we had a couple get-togethers with both the French families and some French researchers. And it was a very exciting trip because after a year of me begging and pestering and emailing her, a French researcher finally agreed to accept KCNH1 to her super comprehensive and impressive $11 million um, uh, potassium channelopathy research program. And it's very cool because they're studying a lot of aspects that we haven't studied for KCNH1 yet. They're making us they've given us dedicated researcher like staff time, and they're making us a zebrafish, KCNH1 zebrafish model, which is, Allie, you can probably say why zebrafish are cool. They're cool because they're transparent, so you can kind of like literally see how, for example, their GI system works or doesn't work and various other things.
I absolutely can. I think they're the coolest research system out there. Um, Zebrafish are an amazing system to model diseases with. They have the same neural cell types and architecture as people do, but they're these teeny weeny little organisms that fit right underneath a microscope. Every time they mate, you get like 200 or 500 embryos, so lots of test subjects, and you can see right through them. So they're beautiful on a microscope. You can do all sorts of genetic tricks with them to help you look at different cell types, and you can test different drugs by just putting them into the water that they swim in. They're really a wonderful organism, so it's really exciting that we're having that developed. And this is a huge accomplishment. This is entirely due to Michelle's advocacy. So great job, Michelle. This is absolutely gigantic that we got into this research program.
Yes, super, super exciting. She was a little hesitant because she's specifically studying potassium channelopathies, which is exactly what our disease is, but the other communities' genes that she's studying have much higher numbers. So she was a little hesitant, but with persistence, she couldn't resist. So and so that these are some of the updates over the past few months. And I was actually just invited to come speak at their channelopathy conference next September. So I will apparently be coming to France again, which is lovely. Um, Allie's going to cover this a bit more later, but we have these very lovely doctor, scientists, clinician researchers, a really wonderful team in Munich that is very interested in studying our registry results. And they got accepted to present the KCNH1 registry results at the AES, American Epilepsy Society. 6,000 neurologists and researchers from all over the world are coming to this conference, and they're going to be sharing our registry results there. They got accepted to present, so that's very exciting. And because of that, they have a hard deadline of, I don't know, I need to ask them exactly when, but we need as much participation with the registry as possible. If you haven't gotten on there yet, Natalia, I don't think I've even talked to you about it yet, but I'll share the link at the end of the session. And it does take a couple hours to work your way through it, especially if you're in a second language, although it is available in most languages, not Polish, unfortunately. But it is a huge, huge contribution to the research and one of the most important things we can do. So if anybody hasn't finished all the surveys, if you haven't gotten on yet, go on, get started. And if you haven't finished all the surveys, try your best to get them all done over the next month or so. We've been talking about this for the past 9 months or so, but Dr. Al George is continuing his CRISPR gene editing project. If any of you saw that exciting story in the spring of baby KJ, who has a rare liver disease and was cured with a CRISPR gene editor, that is exactly the type of treatment that Dr. George is working on for KCNH1. And we had a big deadline to keep we had a big fundraising campaign to keep the experiments on track. And so I reached out specifically to some of the R357Q families for support, and you guys really came through, and we raised $15,000 in like a week and a half. And so that exciting treatment is now about to get studied in mice, which is the first time. We've spent years and years developing our mouse model, and we've spent years and years developing these different, repurposed drugs, RNA treatments, DNA treatments, and now two of them, the gene editors, the CRISPR, and the ASOs, which are RNA treatments, are about to be tested in our KCNH1 mouse model. So that is super, super exciting and has been years in the works. So I'm like literally on the edge of my seat to wait and see how those experiments go. And then finally, I know many of you have been waiting for this. We have these repurposed drugs, like Tristan is already on imipramine. It's only relevant for certain kids if we are sure that your kid has a gain of function mutation. So some of you still need to have that confirmed. We're currently working, some of our researchers are studying some of those variants that we don't know about yet. But probably, hopefully early next year, we will be doing, in coordination with your doctors, if you're interested and your doctor is interested, we will be trying to help some families access these repurposed drugs that, they're not going to be like a CRISPR gene editor, of course, but for Tristan, they definitely have improved some symptoms, and for some kids might have even better results. So it's been a very exciting few months. I'm so happy that, we're back and going to be hopefully meeting regularly again, because we have a lot going on. And I will go ahead and turn it over to Allie now. Oops. Oh, I'll stop and save that for later.
Thank you, Michelle. Great introduction, everybody. It's so great to see you all again. For those of you that don't know me yet, I'm Dr. Allie Rosenberg. I'm your scientific outreach officer, and I am trying to share my screen so I can show you what we're talking about. Here we go. So today we are going to talk about Dr. David Goldstein's presentation, which he gave to us at the same meeting last year that we just brought up, the American Epilepsy Society. His talk was called Bringing Meaningful Therapies from One to Many. And today we're going to talk through this, and we are going to explain a lot of the science that he shows in this today. So let me share with you all his presentation once I figure out how to change my screen. Okay, so as usual, we're going to go through, play some of his video, and then I will explain some of the different topics and themes that come up.
Allie, for some reason, there's a big gray bar at the top. It's not like blocking anything really, but I'm not, and at the bottom.
It's the sharing screen. Hold on. Let me
okay.
Move where my media player is on my screen, and maybe that will be better. Does it look normal now?
Yep.
Great. Okay. Here we go. Let me know if you can't hear it.
I would like to invite Dr. David Goldstein to come up. And he is the co-founder and CEO of Actio Biosciences, and they have a really lovely team of people working really hard there to develop novel small molecules, which are potent and selective channel inhibitors. And I just get the sense that they really understand our sense of urgency as parents and caregivers, and they're really trying to accelerate the time frame to get these treatments to the clinic to help our kids as quickly as possible. So thank you.
So first, let me just say how much I appreciate being here. It's really terrific to have a chance to reconnect with some old friends, some of whom I've known for really much too long to actually state. And it really is a privilege, Michelle and Allie, for us to be part of this very early stage of the process. And it's really wonderful to see a community come together, really dedicate itself to finding an effective treatment. And I will say that KCNH1 epilepsy is a condition that actually should be amenable to an eventual treatment. And so I would say it is well worth a dedicated effort that is represented. I am here with colleagues from Actio in the back. David Breckenridge is our Chief Science Officer. Nicole Miller heads up biology. And thank you for the kind words about the company too. What I'd like to do is just tell you a little bit about how we work in the company to give you a sense of how we approach projects. And then at the end, I'm going to tell you about a little bit of work we're doing specifically on KCNH1. I do want to say that we do not have a full dedicated effort on KCNH1 currently. We would like to at some point, but we don't currently. But we do have some work that we've done and that we are going to share. So it's just a little bit about the way the company works. What we try to do is to pick programs to work on where we know that a gene is responsible for a disease, where we know how mutations in the gene cause disease, and where we believe that it's possible to develop a small molecule, so a tried and tested drug development approach that can counter the effect of the disease-causing mutations, and in that way represent a targeted treatment for what's wrong. And what we do is really scan through the whole universe of genetic diseases to try to find these kinds of opportunities. And once we have found an opportunity, we basically try to convince everybody that's responsible for funding the company that it's worth working on the opportunity. And then we pick these opportunities that we believe are tractable and we can come up with a very effective treatment and that we are able to work on and can make an appropriate argument that it's appropriate for a company to work on them. I won't say anything more about this, but we actually further think about whether anything we develop for the rare genetic disease itself might have some relevance beyond the genetic disease. And the reason that I just mentioned this here is that for very rare conditions, it can be hard to create an economic argument to put a lot of resource into developing a medicine. But if you had a convincing case that it could be used for the rare disease and elsewhere, you could actually see more work done. And so we think about those kinds of opportunities as well.
Okay, so let's get into what Dr. Goldstein was just talking about. And I'm going to share slides again with you all. Okay, so what makes a company want to make a therapeutic for a rare disease? There's
Elaine, we have the big gray blob again.
Yeah, I'm not sure if I know what to do about that. Maybe I can put it down here, over here. All right, I'll put it over here. Does that still work? Can you see now?
Good to go.
Okay. All right, so what makes a company want to make a therapeutic for a rare disease? There are 3 pillars that we think about in this decision-making tree. There's science, economics, and community and resources. So when a company is thinking about if it wants to make a therapeutic for something, first they need to understand if the science is tractable. So do they understand the underlying cause of the rare disease? In terms of KCNH1, we do, right? We know that human beings have different mutations in the KCNH1 gene. And we know what some of those mutations do, which is cause this gain of function. The channel is overactive, right? So we know the underlying cause. We also ask, does the underlying cause seem possible to change with known approaches? So what we know in the case of KCNH1 is that it's very similar to a bunch of other similar diseases that we are able to treat with different kinds of small molecule drugs as well as ASOs. And so Michelle already talked about ASOs. These are RNA-based therapeutics that we're trying to design in a way that are going to inhibit the RNA so the KCNH1 protein isn't made. This has worked for other similar diseases. We know it could work for KCNH1 as well. Also, other diseases have worked by designing small molecule drugs that would do something to, for example, inhibit the channel itself in its protein form. That's been done for other diseases. It could work for us. So the science, KCNH1 is pretty good on that. We're pretty attractive in that way. The next pillar.
And Allie, just to remind everybody, small molecule means what?
Drug.
Like a traditional medicine.
That's right. Like your ibuprofen, your Tylenol. Those are all small molecules that we take on regular basis. We just call them
Something you can
drugs.
just take orally, unlike a CRISPR gene therapy or a RNA, which would have to be administered typically very differently.
Well, a lot of drugs do have to be given through intravenous or other methods too. So it's not just about administration, but it's about what the thing is. And so these are basically small molecule, tiny chemicals, basically. Everything's made of chemicals, larger conversation, but that's what a drug is. Okay, so getting back into the three pillars, economics. This is pretty important because companies need to make money, right? So the company has to ask itself, can it make enough money from the treatment of this disease? And it can take tens to hundreds of millions of dollars to get a therapeutic from the beginning in the lab all the way through clinical trials. So there's really a lot of investment that needs to be made that companies have to think about. Now, the other part of the economics that Dr. Goldstein brought up is the cause of disease shared with other conditions, therefore broadening the use of the treatment beyond the rare population. So what he's talking about here is when we think about KCNH1, that is a very small number of people in the entire world to treat. But KCNH1 is related to some other things. So for example, there's a lot of cancers that have KCNH1 overexpressed in them. One way to broaden the use case for a KCNH1 therapeutic is to think that maybe something that works to depress KCNH1 function would work not just in people with neurodevelopmental disorders related to KCNH1, but maybe some people with certain cancers might benefit as well. There's also different kinds of therapeutics being developed that work for multiple neurodevelopmental disorders, because the neurons in your brain express lots of different proteins and they're all sort of networked and talking to each other. And so there are certain places where the effects of all these ion channels, for example, might sort of funnel together at a certain point. And if we can find a drug that interacts at that sort of funnel point, it might help all sorts of things that have gone wrong before that. So there's a way to think about the therapeutic as not being good, not just for a few people, but for many. And that really, really helps the economic argument. Now, lastly, there's community and resources. So in resources, also known as tools, there's a bunch of scientific tools that make research so much easier for a company. And these are a lot of things that the foundation has actually been working on developing. So this includes cell lines like iPSCs. We talked about those before. These are the induced pluripotent stem cells that come from the biosamples that all of you actually have the ability to provide to our biobank, and then get turned into stem cells, which then can be differentiated into neurons and other kinds of cells for study. The foundation is
And
also working.
I have to jump in here because I see Daria on the call, and we just had another big win that took way longer than it should have. We have 3 patient families in Turkey who had donated blood samples, and it just took an insane amount of time and persistence from Daria and various people to finally get them shipped to the US, where hopefully Al George and our other researchers at Northwestern will turn them into these tools that Ali is talking about, the iPSC lines that will then be turned into neurons, et cetera, to study treatments.
Which is such great news and so important for KCNH1 research. Thanks, Michelle, for that great update. Another thing the foundation has been working so hard on getting is mouse models. We also talked about preclinical models before, mouse being one of those animals, zebrafish another that we just brought up. We've got 2 mouse models in the works, which is really exciting. These are critical for testing what we call efficacy, which is whether or not the drug actually works on the disease. Another thing that we think about is biobank samples. That's not something that we've worked super hard on yet for KCNH1, but this can include blood, urine samples, and also biopsy samples that would go to our biobank. And these are things that can help researchers study different aspects of the disease. The biopsy samples are something that we will talk a little bit more about, I think, in the future, especially regarding if any children have gum reduction surgeries. that's a kind of biosample that we would love to collect in our biobank for study, because that is something unique to our disease. When we also think about resources and community, we think about disease state knowledge. This is super, super critical. So do we know what kind of variants exist in the human population? Do we know what they do? And do we know about the natural history of disease? So natural history is the specific words talking about for just what happens in the disease without treatment. So if you have someone with a KCNH1 related syndrome, what happens at different ages of their life? We would call that the natural history. And that's actually something that's not extremely well known for KCNH1. And that's why the work we're doing at the registry and with these researchers in Munich and some other work we'll do in the future is so important, because without that, we don't really understand how and when to intervene in the disease. Now, lastly, we think about community. Now, for a rare disease, there aren't that many people that obviously have the disease. And so it's important for outside researchers to understand really who they're working with and what their engagement levels are. They would ask things like, are the members of the community aligned in their needs? So do we know what the people that have the disease are like? What things they most think are important as a part of that disease to treat? Are they easy to work with? And are they engaged and likely to quickly enroll in clinical trials to test potential therapeutics? And that last part is something that we're able to demonstrate in present day. One of the ways that we can show our engagement is by having a large proportion of the known community in the patient registry. And that's a number that we can report to companies to say that we are engaged with most of the community and they've filled out all this information about themselves, and we can share that information so you can understand our disease too. So those are just some of the things we think about. They're extremely important. And let's keep going. Are there any questions at this moment, or should I keep going? Sorry for the long diatribe. All right, let's keep going. You can always put questions in the chat. Michelle will be monitoring that because I can't see it while I'm sharing my screen.
Just a little bit about how work is done once we pick a program. What we do in the biology group that Nicole runs is characterize all mutations that are known to cause disease in simple cellular models so that we can test chemical matter against all of them and address this question that Al was emphasizing about the possibility that different mutant forms of the target might respond differently to different modulators. So we characterize literally all of them when we work on a target. And then after that, we characterize appropriate preclinical models. So here you have a disease-causing mutation in humans that's put into a mouse, and then we actually see what happens when you treat the mouse, exactly the kind of approach that Al described, again, for KCNH1. And then we think a little bit about this opportunity to pursue related larger indications. I'm going to give you just one example of what this actually looks like in practice at our company. So we've been working on one program in the company now since the beginning of the company. This is a gene called TRPV4, where, again, similar to KCNH1, mutations cause disease by activating the channel. And so what we need for a targeted treatment here is a small molecule that can inhibit those activating mutations and move the channel's activity back down to normal levels of activity. And so what we do is develop compounds in the company that we hope have those properties, and then we start testing them. And in this case, we have been able to get to a good compound, and that's illustrated here. If you look at the left-hand side of the figure, what you're looking at is a measure of activity of the compound for the normal form of the channel and for all the mutant forms of the channel that are known. And I won't walk through how to interpret measures of activity, but just to say what the figure shows is that there's very good activity against all mutant forms, meaning the compound can be used to treat all patients regardless of what mutation they have.
Okay, let's pause for a second there and So Dr. Goldstein is talking about a drug that they're developing for a disease called Charcot-Marie-Tooth disease in this slide here. Charcot-Marie-Tooth disease has mutations in an ion channel called TRPV4. And what he's saying in this is that the drug that they've developed at Actio is capable of inhibiting overactivity in all known variants of TRPV4. And it's able to make their activity more like the normal version of the channel. Now, something you're going to see in this is on this graph, it says WT TRPV4. WT, just so you know, stands for wild type. Wild type is scientist lingo for the normal version. So I'll sometimes use the word wild type. I'll sometimes use the word normal. You're going to see this in a lot of scientific presentations, just so you know what that means. Now, what he's trying to say here, what you can see on the left side of that graph is that little gray dot for that wild type or normal TRPV4. And on the right side of the graph are all those little orange dots. Those are all meant to represent different variants of TRPV4. And what they're showing in it is that their drug equally binds to not just the wild type, but all the different mutant variants equally, which is great. It means that it should have similar activity with all the known variants, which means they're able to treat everybody in the entire TRPV4 mutant population, which is really exciting. Now, this is just an example of what they're currently working on. And now the next question is, what does the drug do? How does it help? And so what you're going to see next is going to look a little bit sad. You're going to see a preclinical mouse model of Charcot-Marie-Tooth disease. Charcot-Marie-Tooth disease is a kind of neuropathy. That means that the mouse has a lot of motor, moving problems. And so you're going to see this tiny looking little mouse. It's going to look kind of sick and shaky and have a hard time moving around. And then we're going to see what happens after they add drug. All right, I'm resuming, and here we go.
And then excitingly, if you look at the animal model here, where a disease-causing mutation is in a mouse, may be able to look at this. Well, I'm not sure. Are you able to? Oh, there we are. So, yeah, so it should be playing now, I think. Yes. So this mouse doesn't move very much, so it's sick because of the mutation. And that actually has some consistency with what you see in people. Then we treat with an inhibitor, and the behavior of the mouse normalizes. So what this is indicating to us is that the condition is not completely developmental. After the symptoms are present in the mouse, if you treat with an inhibitor that turns off the underlying overactive channel responsible for disease, the mouse gets better. This is exactly what we're hoping to see when we're looking at preclinical results. It's an indicator that this actually may have benefit in patients too, and we will be testing that in patients hopefully over the next roughly year or so. We understand.
Okay, so let's talk
I
about
just it's, it's so sad to see that, very sick little mouse. But I mean, did you guys see how after they give it the medication, he looks like a normal little mouse. He climbs out of the little tray. And it's relevant to, Allie, there's a question in the chat about, age. And of course, we would assume that the earlier you intervene, the better. But as we just saw, that was not a baby mouse, right?
That was not a baby mouse. So great questions. Let's get into this. The big question that David is bringing up here is, can the effects of a disease be reversed? So what I talked about a minute ago is the natural history of a disease, right? That illustrates what happens to someone that has the disease without treatment. Now, with treatment, there's a couple of different things that could happen with the disease. One, the effects of the disease are completely reversed. We would expect all effects of the disease to go away, and the person is going to look just like someone that is the same age as them that never had the disease. Another possibility is that the disease is just stopped. So the disease course stops and it doesn't get worse, but the person still appears to have damage to their system from prior effects of the disease. The third possibility is that a partial reversal. This is just that there's a whole spectrum of what might happen, that the disease course won't get any worse now that they're being treated, and some aspects of prior damage from the disease might go away, but other parts might still remain. Now, we have no clue what will happen without testing. For every single disease, this is the sort of thing that has to be determined by just going through treatment and then cataloging what happens. Some parts of this we can look at in animal models, just like what Dr. Goldstein showed us. For example, the reversal of all of these issues with the motor movements, with walking. For a lot of people that have seizures, we know that giving them seizure drugs can stop them from having seizures, and they are able to get much better in the different ways that they've had issues before. Now, things like cognition and behavior are really hard to evaluate in mice. One of the things that scientists say all the time is that mice aren't people. We don't know what mice are thinking. We have no way to test what they're thinking. And because of that, we can't evaluate how a treatment for a disease makes them change in the depths of their minds. So that's the sort of thing we're only going to understand when we get to find therapeutics that are safe, look like they're effective in the models that we have, and then we make our way to people and do clinical trials and understand over time how things really change. Any questions in this moment? This is a tough topic.
Allie, so Alexandra is asking for the small number of kids in our community who don't have seizures. As Allie was just saying, seizures, everybody, all the researchers and biopharma people always want to focus on seizures because they're observable, countable, and so measuring intellectual disability improvements is much harder, especially as kids are already naturally developing. But for KCNH1, more so than, like, Allie works with KCNT1, this same company, Actio. Just to share, they're moving very quickly. They have started actual clinical trials for that gene, TRPV4, and they're also starting clinical trials very soon for KCNT1. So they're moving very quickly. But KCNT1, for example, the kids have really out of control seizures, whereas for KCNH1, I think our most pronounced and universal prominent feature is more so the developmental delays, the intellectual disability, communication problems. And so that will absolutely be a huge area of focus for us. It's not, just treating seizures isn't enough. We already have medications that for most of our kids do a decent job, not perfect at all, but we absolutely want to work on the intellectual disability and the developmental delays as well.
That's right. And so there are a lot of things that, again, I'm going to talk about the registry 100 times today. There are all sorts of surveys that are present in our registries that help us understand what is the state of intellectual disability and behavioral issues and other things going on with your kid. That's really what the surveys are for, is to help us understand our population. These same surveys get used in clinical trials to help understand if the therapeutic helps change things and make them better or worse. And so that's what our registry kind of helps us do, is establish something called a baseline. Where are we starting? What is the kid like at different stages in their life? So we have an understanding of that. And then with treatment, we can compare how it changes. So that's what it's really important for. At the start of any trial, you'll see a lot of these same surveys be given again as that baseline measure. So there's a direct comparison later on with treatment, if that makes sense. But yeah, seizures isn't everything. It's one thing that a lot of neurodevelopmental diseases share, and it's an sort of easy endpoint because it's so visible. So a lot of companies like to use that. For KCNH1, it's not going to be the focus of any therapeutic program. They would look at other things for us. Okay, ready for me to keep going? All right.
what about KCNH1? So before I share results that we do have for KCNH1, let me just say that this is not currently a dedicated full program in the company, and we don't know that it will become a dedicated full program in the company. But we do at the company operate on a philosophy of being transparent about what we do. So we're going to share where we are now, but we don't want that to be taken as any kind of a promise that we're going to be able to keep doing a lot more work, because we don't know what we're going to be able to do yet. But we do.
Okay, I wanted to pause again because this is a really important point. David is going to be sharing with us, not just the results of his TRPV4 treatment, but also some early results for a KCNH1 program. And what is really important for us all to remember is what he just said. He's not making us a promise that he is definitely going to pursue KCNH1 all the way towards clinical studies. Like I said at the beginning, and I'll say this again later, there's a lot of things that we have to do to kind of be able to get on the list for a pharma company to be able to make them want to put in the enormous multi-year and multi-million-dollar investment to develop something for us. So he's helping us and he's doing some work right now, but not a promise. And I just wanted to make that really clear that everyone understood that. But there's a lot of great things happening so far. So let's keep going.
You have an extremely strong chemistry team, and they're really, really good at looking at all the compounds that are known to have some degree of activity, and in a process I don't understand at all, having an inspired guess about compounds that they could make that actually might have improved properties. And they've done some of this chemistry magic, thinking about inhibitors for KCNH1, and this is where they have gotten to in the team. So I'm going to move maybe just a little bit. If you look at the left-hand side, you'll see imipramine. That's something I know that has been tried, and we actually have a measure of how active it is against KCNH1, and you can see that that number is very large. The way to interpret these measures of activity is the larger the number, the less active. And for imipramine, it's a pretty big number, so the activity against KCNH1 is actually pretty weak. Abilify, similar things. These are two medicines that are used for psychiatric conditions. Again, number is pretty large and the activity is pretty weak. So what that actually suggests is that it would be difficult to take enough to have meaningful inhibition of KCNH1 without doing a lot of other things that you don't want to do. And I will just make one quick comment. I am fully supportive of the kind of repurposing efforts that Al was talking about and a lot of people work on and that I myself have worked on. And I think we should carefully look to see if any approved medicines can help. I fully believe in that. But it is also my experience that normally you do not find anything approved that works well enough. And that's why you need a dedicated drug discovery effort to improve upon what compounds need to do to make them fit for purpose. So let's explore repurposing, but we also almost always need to improve. Compounds.
Okay, so let's talk a little bit about therapies targeting KCNH1. All right, so what Dr. Goldstein was talking about is that Actio is searching for a compound that is potent and selective for KCNH1 mutations. So potent, it's strong, selective, it is specific, while minimizing the potential for side effects. And the reason for this is that we know from, the last 100, 200 years of history that lots of central nervous system drugs are not very selective. And they're active against multiple different kinds of receptors and proteins, and that can lead to side effects. Now, in KCNH1, the thing that we worry the most about is its cousin, which is called hERG. hERG is found in the heart, and the issue is that it's really similar to KCNH1, and if things that drugs that we're adding bind to KCNH1, it may bind to its very similar partner, hERG, and then cause issues in the heart, specifically proarrhythmia, which would be a long QT syndrome. that would be terrible for our community. We really don't want to see that happen. So this is why safety testing is so important, to make sure that we're getting good separation drugs that combine to H1, but not HERG. So let's talk a little bit about our repurposed drugs here. So what Dr. Goldstein was saying is, when you look at these potency numbers, high numbers, and we're in the thousands here, mean that they're not that selective. They're not that great. That means that they're not that great at binding just to KCNH1. So both imipramine and Abilify have pretty high numbers. Still means that they bind to them, but it's not the only thing that they're going to bind to. Now, what you can see for imipramine,
it's the best thing we know about right now, but it's certainly not perfect.
it's not perfect, and we're going to talk about this. So HERG, you can see, has a pretty similar number to imipramine, which kind of means it might sort of hit both of them, depending on how the drug is moving through the body. And Abilify has a lower number for HERG, which means it's actually more specific for HERG than for Abilify. But there's some really important caveats to this information. This is the wild type, or the normal version of the channel. Now, what we're talking about in our community is not the wild type channel. We're talking about our different variants. And what we've talked about in previous explainer seminars is that our variants change the protein. We all know that it's already working differently. What I can tell you at a molecular level is that it's also probably shaped differently and acting differently in different ways. And these numbers have not been evaluated in variants. Sometimes drugs can bind much better to variants than they can to the wild type channel. We just haven't looked at it yet. We don't exactly know the answers to this, but it's something that we need to understand. And so what we're going to do to figure this out is we're going to launch what we call observational studies, where we're going to set up a way for everyone to get information about these two drugs, how to take it. We're going to build in lots of cardiac safety checks so everyone understands how to take these safely and what to look for. And then we're going to set up a way for everyone to document how they look, so we can record and understand, do they make a difference? Because there's a good chance that these drugs may actually be helpful for some members of our community. And the only way to figure it out is to try it. So that's something that we will roll out. We'll do it as safely and carefully as we can.
And Allie, I believe that both Luis Pardo and Al George have screened either one or both imipramine and Abilify, and confirmed that in some cases they do bind more potently to our kids' mutated KCNH1 proteins, to our kids' unhealthy channels, that they actually work more strongly against the unhealthy channels than the healthy, and help correct the function of the channels.
Which is how they were found and why we think they're useful to try. What's also really important is that these compounds have both been tested for safety in children before, and they really are safe in your average person. So again, we don't know for our population, we're obviously different than your average person that has a healthy KCNH1 and a healthy HERG. So we'll find out, and we'll talk about more about this once we've got our system in place. Okay, I'm going to keep going and share the rest of this talk to the end.
That have been made in the company with a small chemistry effort are indicated on the right-hand side. And what you can see is these numbers get pretty small, showing dramatically increased activity against KCNH1 in comparison to what's available. Now, these are not ready to be used. These are not drugs. These do not have drug-like properties. We haven't even characterized what the properties are like. But what we do know is that they have meaningful activity against KCNH1. And to me, what this is indicating is that this looks possible. The other thing that I would really emphasize is that the work that's being done is starting to show some separation of activity between activity against KCNH1 and activity against that heart channel that's closely related, that has been referred to many times. There, you want to be very careful about not having activity. They are closely related, the heart channel and KCNH1, but you can see that we have compounds that begin to separate activity against KCNH1 and the heart channel, indicating that it is possible to separate. So the message of all of this to us, we are a long way from something that can be used in people, a very long way, but it is starting to look like it is possible to do with a dedicated effort. So the final thought that I'll leave you with is that the more we understand the biology of KCNH1, the easier it is to motivate serious drug discovery efforts. One reason for that is it may give us pointers that there's relevance elsewhere that'll really motivate us and others to work harder on this program. We're going to try to work on it anyway. I think people who know me know that. But the more we know about it, the more motivation there will be to work on it. And there are some directions that can be pursued you've already heard about earlier, such as trying to understand what cell types are the most affected by KCNH1 mutations, and then actually seeing how modulators of KCNH1 affect that. We actually think we probably won't be that far away from having a tool compound that could be used in those kinds of experimental settings to see what happens in different kinds of neurons, to see what happens when you put it into a mouse. And I'll just say, since many of the leaders of this kind of work are here, that this is work we'd love to collaborate on and help in any way that we can. And that's really what we wanted to share, and it really is a pleasure to be here. Thank you.
Okay, so let's talk about the last part of his presentation.
And one quick note, as he mentioned, many of the leaders in the field of channelopathies were there at that meeting, and he said we're looking for collaborations, and we were able to set that up there. So they have continued working on these KCNH1 compounds over the last 9 months since we were at that meeting. And due to meeting up with Dr. Al George there at that meeting, Dr. George also did a presentation. We were able to connect to the two of them, and they are now currently in the process, or hopefully they've actually arrived. They have shipped these exciting KCNH1 compounds to Dr. George's lab, and he will be screening them in all of those different mutations, different variant cell lines that he already has up and running. So again, that's, kind of the beauty of being a patient advocacy group, where we bring all the people studying the disease together, and they can save so much time and money by sharing these tools.
Absolutely. And Michelle's done an incredible job in facilitating those relationships and keeping the Actio team really motivated. So once again, amazing job. So let's talk a little bit about these compounds for a second. We talked a little bit before about how high numbers mean not great binding, but low numbers mean great binding. And these numbers are way less than in the thousands. They're in the hundreds and below. So we can see is that these low numbers mean that they're binding to KCNH1 nicely. And you can see that the numbers are higher for hERG, which means they're less likely to bind to hERG, which means you could take a smaller dose of these compounds and they will preferentially bind to KCNH1, which is really exciting. That's what he meant by separation. We're getting a preferential binding to KCNH1 and not to hERG. Now, this is what David was talking about, thinking about a precision treatment. So this is the best kind of potential therapy for KCNH1. There's lots of other kinds of things that we're working on too, like ASOs that are other kinds of precision treatments. And these kinds of things have been tested in other neurodevelopmental disorders. So we're really excited that we're moving in the right direction, that David is collaborating with other researchers. And like he said, there's a lot to still understand about KCNH1. For example, what kind of neurons are most important to try to treat, as well as to show our community engagement that helps keep them excited and motivated. I also want to, get into our little summary here, which is that, before we get into the summary, also there's other treatments being developed that are not precision treatments for KCNH1. However, they're sort of in that funnel effect of lots of different things can go wrong in neurons, but those effects all sort of funnel down, and we might be able to treat at that spot where they all come together. One of these treatments is called bexicaserin. It's developed by a company called Lundbeck, and they are currently enrolling for clinical trials. This is a kind of drug that isn't designed for us, but it might still be helpful. And one of the inclusion criteria for this trial is that your child needs to have 4 or more motor seizures per month. So I know that we're not focused on seizures in this community, but if your child is having 4 or more seizures a month, let us know and we can provide you more information about this trial in case you might be interested in trying this therapeutic out. And as we learn about things like this, the foundation will continue to share them with you all, give you the right information about what is a good idea to try out. So, in summary, Actio is working on a test compound that can be used in lab experiments, but not people at this stage. They think that the disease caused by KCNH1 is very likely to be improved with treatment, but again, degree of improvement is something that we won't know without testing. And that our community has a role to play. We can demonstrate to companies that we are a good target to work on by showing our engagement. The most important thing above everything is signing up for the registry and filling out those surveys. We're going to keep talking about that. If you're in the US, please sign up for the Citizen platform. That is about a 5-minute sign up, and Michelle will talk more with the US families about that. But that's something really exciting. I'll tell you about that in a second. You can help with fundraisers. You can share your story. All of these things demonstrate to outside researchers and companies that we've got an engaged community that really wants investment. And really importantly, on the research side, the Cure KCNH1 Foundation is working so hard to invest in and support the development of all kinds of research tools, cell lines, mouse models, testing human variants to understand how they work. All these things are so critical, as David mentioned, to help us move forward. So let's talk more about the registry. The reason that the registry is becoming even more important is that we have engaged these wonderful researchers at LMU in Munich, Germany, and they're really excited to study KCNH1, and they applied to and were accepted to present information about KCNH1 at the biggest neurology conference in the world. That's AES. It's in December, which means that they need time to analyze our registry data and write it up for this conference. If they don't have results by the end of October, they're not going to be able to include them in their presentation. So this is the month to really engage with the registry. What's really important about the presentation that they're going to give is that this is the kind of thing in front of a huge audience that has the possibility to attract a lot of interest. They're going to talk to the world about KCNH1, and that can help other researchers want to research it. It can help pharma want to think about developing therapeutics. It can help funders want to think about getting involved in our cause. So we're going to ask you all in October, this October, just to fill out 1 hour of surveys. And that 1 hour of surveys can answer their most pressing questions. And Michelle will talk to you more in a second about those. Also, if you're in the US, please enroll in Citizen. It takes about 5 minutes. And we were really excited to tell you all, we've won a grant from Citizen to help us study this data. Citizen is a company that grabs electronic medical health records from all your different doctor's offices and aggregates them in one place. This is the kind of information that we can use along with our registry to put together what we call a retrospective natural history, where we can analyze information we already have to explain our population. As many people as possible should enroll. If you're in the US, you are eligible. Oops. And so for the registry, what we are asking you to do, and Michelle is going to follow up about this, is to upload your genetic report and just fill out a handful of surveys. First, you have to fill out the health and development survey. This is sort of the intro, yes, my child has this, no, my child doesn't have this survey. It should only take probably 10-ish minutes. And from there, it opens up a bunch of other follow-up surveys. You should next do the brain and nervous system survey, and then seizures and development 1 and development 2. That is less than an hour of clicking buttons. Matrix does work on your cell phone, so it's the sort of thing that you could do while you're watching a show. So please engage, please do this, less than 1 hour, and it'll make a huge, huge difference for what we can present to the entire world this December. And if you're feeling motivated, there's other things those researchers requested. It will take you more than an hour to do them, but that'll be all that we ask from you this October. So I'm going to stop sharing And I'm available for questions. Michelle is available for questions. I don't know if we have time to log into Matrix together and see if that's working, but Michelle, the floor is yours.
Well, I mean, you basically said everything, but we're under a time crunch, and the more surveys that, everyone can fill out, the better this month. And so I put in the chat a clickable link for the registry. Many of you have already filled out everything, but you might just want to double check and make sure your genetic test result is in there, because that's a crucial core piece of information. As Ali said, we just launched this earlier this year. It's a work in progress. I'm going to go to the company we're working with for the platform and try to make it more clear which surveys are super important, the ones that Ali just talked about. But for right now, if you can just log in, make sure you have your genetic test results in there. And then the way it's set up, it's, again, I'm going to try to make it a little more easy to access, but you have to fill out the health and development survey first. And then you might think, oh, I've done everything. And then a whole bunch more surveys get added based on your responses. So like if you say, yes, my kid has the gum problems, then or like, yes, my kid has developmental delays, then the developmental ones get added. If you say my kid has gastrointestinal issues, then the GI one gets added. So you might have to like log in a couple times, fill out the health and development one, and then wait for all the other ones to get added, then fill out the brain and nervous system one. Once you fill that out, then the seizure one gets added. And it's unfortunate that it's 3 steps in. I'm going to try to change that as quickly as possible, but right now you might have to like log back in a couple times as things get added as you make your way through them.